Everyone, we are the Group 3 and we are assigned to discuss about the benefits and risks of using GMOs or genetically modified organisms. By the end of the discussion, the students should be able to articulate the principles of genetic modification, identify the benefits and risks of genetically modified organisms, analyze the societal and environmental implications of GMOs, and evaluate the ethical considerations surrounding their use in various fields.
Unlocking of difficult terms. Number 1. Gene. A gene is a segment of DNA that contains instructions for development and functioning of an organism. In GMOs, the specific genes are added or modified to introduce desired traits. Number 2. Recombinant DNA. A recombinant DNA is formed by combining DNA molecules from different sources. In GMOs, this technique is used to insert genes from one organism into a genome of another. Number 3. Selective breeding. Selective breeding is a traditional method that involves choosing certain organisms with the desired traits to reproduce and create offspring with those traits. While not considered, genetic engineering is a form of genetic manipulation. Number 4. A resistance. A resistance refers to the ability of an organism to withstand or repel certain traits. In GMOs, genes are inserted to make the organism resistant to pests, diseases, or herbicides. Number 5. Ethical consideration. The use of GMOs raises ethical questions such as potential environment impacts, ownership of genetically modified seeds, and the potential of unintended consequences.
What are genetically modified organisms? A genetically modified organism or GMO is an organism whose genetic material has been altered using genetic engineering techniques. Organisms that have been genetically modified include microorganisms such as bacteria and yeast, plants, and mammals. Source of genetically modified foods and are also widely used in scientific research and to produce useful goods other than food. So in other words, genetically modified organisms refer to a plant, animal, or microbe in which one or more changes have been made to the genome, typically using high-tech genetic engineering in an attempt to alter the characteristic of an organism. Examples of genetically modified organisms are alfalfa. GMO alfalfa is primarily used to feed cattle, mostly dairy cows. Most GMO alfalfa is resistant to herbicides, allowing farmers to spray the crops to protect them against destructive weeds that can reduce alfalfa production and lower the nutritional quality of the hay. Apple. A few varieties of GMO apples were developed to resist browning after being cut. This helps cut down on food waste as many consumers think brown apples are spoiled. Sugar beet. Sugar beets are used to make granulated sugar. More than half the granulated sugar package for grocery store shelves is made from GMO sugar beets. Because GMO sugar beets are resistant to herbicides, growing GMO sugar beets helps farmers control weeds in their fields. Pink pineapple. The GMO pink pineapple was developed to have pink flesh by increasing the levels of lycopene. Lycopene is naturally found in pineapples, and it is the pigment that makes tomatoes red and watermelons pink. We are now in principal objective of production. The basic principle for producing a GMO was to add new genetic material into an organism's genome. This is called genetic engineering and was made possible through the discovery of DNA and the creation of the first recombinant DNA molecules by Paul Berg in 1972. Transgenic. Transgenic plants have been engineered for scientific research to create new colors in flowers and to create improved crops. In research, plants are engineered to help discover the functions of certain genes. One way to do this is to knock out the gene of interest and see what phenotype develops. Another strategy is to attach the gene to a strong promoter and see what happens when it is overexpressed. So, genetically modified plants are those that have been genetically modified using recombinant DNA technology. Genetically modified crops. In agriculture, genetically engineered crops are created to possess several desirable traits such as resistance to pests, herbicides, or harsh environmental conditions, improve product shelf life, increased nutritional value or production of valuable goods such as drugs or farming. Plants including algae, jatropha, maize, and other plants have been genetically modified for use in producing fuel known as biofuel. The purpose of genetically modified crops generally include resistance to certain pests, diseases, or environmental conditions, or resistance to chemical treatments. For example, resistance to a herbicide. Other purpose of genetic modification of crops is to enhance its nutritional value as seen in the case of the golden rice. Golden rice is a new type of rice that contains beta-carotene or provitamin A plant pigment that the body converts into vitamin A as needed. This compound is what gives this grain its yellow-orange or golden color, hence its name. Golden rice is developed through genetic engineering. While ordinary rice does produce beta-carotene, it is not found in the grain. Thus, scientists used genetic engineering to add the compound to the grain, a minor tweak that improved grain's nutritive value. The beta-carotene in golden rice, which was made possible by the addition of two new enzymes, is identical to the beta-carotene found in the green leafy and yellow-colored vegetables, orange-colored fruit, and even in many vitamin supplements and food ingredients.
Hello everyone. Now I will be discussing about microbes. But before that, let me ask some question. What do we mean by microbes? Microbes, also known as microorganisms or microscopic organisms. These are the tiny things that are found all around us and too small to be seen by the naked eye. So, it means ang microbes daw, ito yung mga mikrobyo sa pagkakaalam natin na hindi basta-basta nakikita ng dalawang mata natin. Kasi sobrang liliit ng mga mikrobyo na kailangan gumamit tayo ng microscope para makita yung itsura ng mga microbes. Microbes can be bacteria, fungi, archaea, or protists. Now let's discuss about microbes. Bacteria were the first organisms to be modified in the laboratory due to their simple genetics. So, malino naman ang pagkakasulat na ang bacteria daw ang unang organisms na na-modified sa laboratory dahil sa simple genetics ito. These organisms are now used for several purposes and are particularly important in producing large amounts of pure human proteins for use in medicines. So, ang organisms ng microbes nakatutulong or useful sa medicine kasi ang I bet sa pagkakaalam natin ng lahat, 'di ba, ang mikrobyo nagdadala sa atin ng sakit like ng sipon, yan, yung ubo. But kung titingnan natin yung other side ng microbes, nakatutulong din ito. Kasi sabi rin dito, genetically modified bacteria are used to produce the protein insulin to treat diabetes. Yan, so hindi rin ganun kasama ang mikrobyo. Similar bacteria have been used to produce clothing factors to treat hemophilia and human growth hormone to treat various forms of dwarfism. So, it means yung nabanggit na bacteria or yung similar bacteria, hemophilia, ito yung bleeding disorder in which the blood does not clotting properly and can cause abnormal bleeding. So, means, ang hemophilia daw is yung pag-stop ng dugo sa pagka-clotting niya. And then, yung dwarfism from the word dwarf. Alam naman natin ang dwarf is maliit yung size nila, diba? Dwarfism, short stature that results from a genetic or medical condition. An adult height of 4 feet 10 inches. Yan, yung mga tinatawag nating little person. Yan. Sila yung mga taong nahinto yung pag-growth ng height nila. Like, for example, ang age niya is 36 years old pero ang height niya is pambata. Sila yung mga tinatawag nating dwarfism. Next, let's talk about mammals. Lahat naman tayo siguro familiar sa mammals, di ba? Mammals, these are living things that are warm-blooded vertebrates, and they feed their young with milk from special mammary glands of the mother. Yan. Ano ba examples ng mga mammals? Siyempre, unang-una dyan tayo, mga humans. Ano pa? Like, ito, yung sa picture, giraffe, dog, dolphin, cat, elephant, what else? Cow, and etc. Ralph L. Brinster and Richard Palmiter developed the techniques responsible for transgenic mice, rats, rabbits, sheep, and pigs in the early 1980s. So, short background check tayo kay Brinster and Palmiter. So, si Ralph L. Brinster, he studied animal science and undergraduate at Rutgers University, while Richard Palmiter, he's a professor of biochemistry and genome sciences. He is also known for his contributions to 4 different areas of molecular biology and animal physiology. They established many of the first transgenic models of human disease, including the first carcinoma caused by transgene. So, when we say carcinoma, ito yung cancer that forms in epithelial tissue. So, nag-research ako about dito, and then ang nabasa ko, ito daw yung first, parang pinakaunang cancer. Hindi siya yung stage 1 cancer, ha. Like, yung parang bago yung cancer, nauna tong carcinoma. Yan. And then, the process of genetically engineering animals is a slow, tedious, and expensive process. However, new technologies are making genetic modifications easier and more precise. So, sa pagkakaintindi ko is siguro nung mga dun sa past is yung ang proseso ng pag-genetic engineer sa animals is mabagal at saka may kamahalan, pero ngayong modernize na yung mundo natin, marami ng technologies na naiimbento na nakatutulong yung mga technologies na ito para mas mapadali yung pag-genetic modifications sa mga animals. Next, production of genetically modified organisms. Yan. Genetic modification involves the insertion or deletion of genes. So, alam ko lahat na tayo familiar sa sa word na insertion or at deletion, 'di ba? Sa science, sa DNA, yan. When we say insertion, from the word insert, adding one or more nucleotides dun sa gene, while the deletion from the word delete, removing at least one nucleotide in genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. Yan. In nature, this can occur when exogenous DNA penetrates the cell membrane for any reason. Next, agrobacterium ability to transfer genetic material to plants, or the ability of lentiviruses to transfer genes to animal cells are natural examples of gene transfer. So, when we say agrobacterium, ito yung genus of gram-negative bacteria that uses horizontal gene transfer to cause tumors in plants. So, ang agrobacterium, hindi natin masasabi na good or bad ang agrobacterium kasi it's just trying to survive. Pero dahil sa pag-iisip ng mga humans, dahil sa cause or sa effect ng agrobacterium sa plants, na nagkakaroon ng negative thinking yung mga humans about dito. But there's hindi siya specific na good or bad. Yan. Also, yung lentiviruses, ito yung genus single-stranded RNA viruses such as HIV and SIV. Yan. And then, nagkakos din to ng fatal sa human at saka diseases sa animals such as AIDS, equine infectious anemia, and ovine progressive pneumonia. Yan. And next, these are the intended purpose for genetic modification of mammals. First is to research human diseases. Second, to produce industrial or consumer products. Third, to produce products intended for human therapeutic use. Next is to enrich or enhance the animal's interactions with humans. And lastly, to enhance production or food quality traits. So, these are my parts of our report, and I hope you learn something from my discussion. That's all. Thank you.
Genetic modification involves the insertion of deletion of genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. In nature, this can occur when exogenous DNA penetrates the cell membrane for any reason. Agrobacterium ability to transfer the genetic materials to plants, or the ability to lentiviruses to transfer genes to animal cell are a natural example of gene transfer. Intended purposes for genetic modification of mammals. To research human diseases, for example, to develop animal models for these diseases, to produce industrials for consumer products, fibres for multiple uses, to produce products intended for human therapeutic use, pharmaceutical products, or tissue implantation, to enrich or enhance the animal interactions with humans, to enhance production or food quality traits, faster growing fish, pig, that digest food more efficiently.
Engineering, a groundbreaking scientific technique, has been revolutionizing numerous fields, including medicine, agriculture, and industry. It involves the direct manipulation of an organism's genes, often incorporating DNA from one organism into another, to create or enhance traits. The term genetic engineering is generally used to refer to methods of recombinant DNA technology, which emerged from basic research in microbial genetics. The technique employed in genetic engineering have led to the production of medically important products, including human insulin, human growth hormone, and hepatitis B vaccine, as well as to the development of genetically modified organisms such as disease-resistant plants. Genetic engineering is a process that alters the genetic makeup of an organism. This can involve the introduction of foreign DNA or synthetic genes into the organism of interest. The primary aim is to introduce new characteristics or traits which the organism does not naturally possess. The technique has been used to create genetically modified organisms or GMOs that exhibit traits such as increased resistance to pests and diseases, enhanced nutritional content, and improved tolerance to adverse environmental conditions. These GMOs have significantly contributed to the global food supply, addressing issues of food security and agricultural sustainability. In-depth exploration of how genetic engineering is used in various fields.
Traditional breeding. Modification of plants and animals through selective breeding. Practices used in traditional plant breeding may include aspects of biotechnology, such as tissue, culture, and mutational breeding. Traditional breeding involves selecting and mating plants or animals with desirable traits to pass those traits to the next generation, a method used for centuries in agriculture to improve crops and livestock. Examples of traditional breeding include crossing different varieties of plants to enhance disease resistance, increase yield, or improve taste. In livestock, breeding is done to achieve traits like better meat, quality, or increase milk production.
What is pros? Pros is increased attractiveness to consumers. For example, apples and potatoes that are less likely to bruise or turn brown. 2. Enhanced flavor. 3. Longer shelf life and therefore less waste. 4. Greater resistance to viruses and other diseases which could lead to less waste and increased food security. 5. Greater tolerance to herbicides, making it easier for farmers to control weeds. 6. Greater tolerance to herbicides, making it easier for farmers to control weeds. 7. Increased nutritional value, as in golden rice, which can boost the health of people with limited access to food. 8. Greater resistance to insects, allowing farmers to pesticide use. 9. Ability to thrive in a harsh climate such as drought or heat. The last number 10. Ability to grow in salty soil.
Allergic reaction. There is a small risk that GMO foods can trigger an allergic reaction, but this will only happen if the genetic change trigger the production of an allergen. The risk is small, but there is a possibility of triggering an allergic reaction if GMO foods produce allergic substances. Cancer. There have been concerns that eating GMO foods can contribute the development of cancer by rising levels of potentially carcinogenic substances in the body. Some concerns have been raised that GMO foods may contribute to cancer development through potentially carcinogenic substances. Antibacterial resistance. Some GMOs contains changes that make them resistant to certain antibiotics. In theory, the genes from these planets could enter humans or animals when they eat them. As the result, the person or animals could also develop antibiotics resistance. Consumption of GMO's food may lead to antibiotic resistance in humans or animals. GMOs resistant to antibiotics could transfer their genes to humans or animals upon consumption, potentially leading to the development of antibiotic resistance. Changes in human DNA. Eating GMO foods could lead to genetic changes in humans. However, most of the DNA in foods, whether GMO or not, either is destroyed by cooking or breaking down before it reaches the large intestine. While GMO foods may lead to genetic changes in humans, most DNA in foods is destroyed by cooking or breaks down before reaching the large intestine.
That's all. Thank you very much.